before, is the cortical wiring and receptive field properties of cortical cells, this crystalline cortex that he's talked about, the units that are repeated in the cortex. And what we asked him to talk to us about today is the development of this system, and in particular, the development of binocular abilities. And so he's going to talk to us about that today. This was part of the work that the Nobel Prize was originally based on. The other thing I wanted to announce, this was sort of in progress last fall, and is very special for us, is that David actually has accepted an appointment as a distinguished visiting fellow in neuroscience here at Wellesley. So he's actually a little bit one of us now. He has been so incredibly generous with his time and his energy. What? Yes, I'm getting there. Okay. We have trouble with that, yeah, hieroglyphics, whatever. Yeah. Anyway, so he has been giving multiple lectures here each year, and for the last couple of years, and has also even gone to some of our Intro Neuro 100 labs to help. He actually gave us many of the ideas and some tutoring for how to create one of those labs. And something else that many of you may know is that because he refuses to accept an honorarium, he just comes here because he loves us, I guess, and loves telling us what he's done. In lieu of these honoraria, we've been able to create a first and second year writing prize in neuroscience. Which do you have a deadline for this spring?
You can see the posters around it sometime in April.
Okay. So this is some kind of original writing work. It can be a poem or a story, or there was a rap last year that actually was one of the 3 finalists. And so it can be any piece of original work. Last year, Bevel, Conway, and David judged these, and then all of the neuroscience faculty got to read them and endorse those decisions. And so this is an incredibly special thing. David's a wonderful writer, and so for him to establish a fund to fund this writing prize is really incredibly terrific for all of you. And so anyway, thank you, David, for being here and for all that you've been investing in us recently. And these guys obviously appreciate it. They're out now in the hallway going to listen to you. So anyway, welcome, and I'm going to just turn the floor over to you.
Okay,
Yeah.
Thank you.
What do you think about the lights? Maybe just the front down, because the slides won't be as all that bright?
Yeah, we can do that.
Just one and then
Yeah, that's good. It was just the other day that Bevel told me that I should talk about deprivation, which was a surprise to me. I don't think I've talked about this subject in a lecture for the 10 or 15 years, which is recent as far as I'm concerned. But I had to go to some of the original papers and to the book I wrote to try and refresh my mind. My one problem that I have is that I don't know how much you people know. And so I'm going to start sort of early in the game and cover some ordinary physiology and anatomy before I get to the development and deprivation work. The background of the deprivation work, Torsten Wiesel and I started studying development and deprivation around I guess it was in 1963 or something like that. In the first 3 years that we collaborated, we thought that we had learned enough about how the visual cortex worked that we could do some simple experiments to try and get at the question of how the whole thing developed. And development you can divide into 2 segments. There's prenatal development, how much of it there comes when the animal is born all ready to work, and how much of it is developed through, let's say, something loosely speaking that we might call learning. How important is experience in the wiring of the brain? So we did our first experiment in this, and it was nice in those days. You didn't have to write a grant request or ask permission or fill out forms. we just decided that we would take a newborn kitten and sew the eyelids of one eye closed under an anesthetic, of course, let the animal recover, and let it go for a period we decided arbitrarily on 3 months. I don't know why we decided on that length of time, but after 3 months, we anesthetized the animal again, undid the eyelids, and recorded. And so the first experiment was done in order to see how severe, how disastrous an effect on the animal's visual system did closing the eye result in. So that's the background. But in order to refresh your memory and mine a bit, let me just go through some of the nuts and bolts of the visual system in mammals. And the animals we work with are cats and monkeys. For a while we used them interchangeably. Finally we graduated, and in the last 10 or so years of collaborating, it was monkeys only. But the original work was done in cats, and what amazed us along the way was how similar the visual cortex was in cats and in monkeys, which indicated to us that probably the brain, the visual part of the brain in us is probably rather similar to that in monkeys. Because if monkeys are like cats, then a fortiori probably monkeys are like us. Anyhow, the first thing that one wants people to learn is the wiring of the eyes to the lateral geniculate bodies and hence to the brain. And the first slide, when I have the slide, that I always show in every talk practically that I give is a diagram of this system. The slide was at the lab, and it snowed so hard yesterday that I didn't get in, so I don't have the slide. I have to draw it on the board. And if these are the two retinas, with the lens here, and the scene out here, if you have a cell, a retinal ganglion cell with its optic nerve fiber coming in, it comes in to the optic chiasm, and if it originates on this side of the eye, it stays over here. And similarly, something coming here to the chiasm ends up like that. Something coming here crosses to the other side, and this side comes here. And this is the lateral geniculate body, where the optic nerve fibers end. Now, this is probably something that you all know. I used to tell medical students that this is a diagram that they should be able to draw even if they were blind drunk. And then in one lecture that I gave to medical students, I told them this, and then I proceeded to get it all wrong when I drew it on the blackboard, which was embarrassing. The other part of the story is that we put this on an exam once for medical students, and one poor student got it all wrong, so he got the fibers from here crossing to this side, and everything was completely scrambled. But then he forgot that something out here in the environment projects to this side of this retina and this side of this retina. He got that all wrong. So the result was that it came out right. He got, and so we had to give him full marks for this. And my one major regret in teaching medical students is that I didn't keep that exam question. It would have been wonderful to show to the students, but alas, I must have thrown it out. Anyhow, this is what you need to know. So, and of course, the postsynaptic fibers go to V1, primary visual cortex on both sides, and V1 on the two sides are interlinked by the corpus callosum. And so you can go on and on with the basic circuit. This is the main circuit that you need to know even if you're blind drunk. So what I want to do is start by showing you what the lateral geniculate body looks like anatomically, and then we'll go on to the cortex, look at it anatomically, and describe how the cells act. What you must surely know by now is that optic nerve fibers and lateral geniculate cells, if you look at their receptive fields, that is how the world, information coming from the world affects these cells. The receptive fields you can describe as being center surround, so that if we're looking at the retina and you shine a spot on the center, you may make the cell fire faster. If that is the case, then when you shine it on the surrounding region, you make it fire slower or stop it from firing. That's one typical type of retinal ganglion cell. There are really two basic types. This is one type with plus in the center and minus in the surround. The second type is the reverse. And this is discovered in around 1950 by Stephen Kufler, who ended up the chairman of our department, and we joined his lab at Hopkins, and everybody moved up from that lab in 1958 to Harvard. So this is the main work of Kufler, and it was certainly a surprise to everybody to learn that the cells worked in this way. What is interesting about this, and I don't think this was even fully realized by Kufler himself, and it was quite a while before we realized what the significance of this was. And it turns out that if you take a typical retinal ganglion cell, whose axon is the optic nerve fiber, if you record from it, and if it's a center surround on-center cell, if you bathe the whole receptive field in light, if you use diffuse light to stimulate, you're telling the cell to fire faster and slower at the same time. It tends to sit there and do nothing. So diffuse light is not an adequate stimulus for a cell like this. In fact, with optic nerve fibers, usually the center wins out over the surround. But for many cells, it's very close to a draw, and you get no response at all with diffuse light. So to make a cell like this fire, you have to, if you shine light on it like that, you'll tend to inhibit the cell strongly because you're shining light on a fair portion of the surround and none of the rest. If you shine light like this, you're shining light on all of the center and only half of the surround, roughly. So that will make the cell fire faster. So these cells tend to respond to contours. They're not sitting there asking whether there's a spot of light somewhere in the environment. It's contours mostly that they're interested in. And they're not interested in diffuse light, at least not completely. You know that these cells respond to diffuse light simply by shining a flashlight into the eye of a person. The pupil will constrict, which must tell you that some retinal ganglion cells do respond to bathing the whole retina in light. And that is simply because the center for those cells is won out over the surround. So that's a brief summary of the workings of both retinal ganglion cells, that is the output cells of the retina, and the lateral geniculate cells. It turns out that one thing you can ask if geniculate cells work roughly the same way as retinal ganglion cells, what is the geniculate there for? And we know, at least in the cat, several things about what the geniculate does. One is that it beefs up the power of the surround to blank out what the center is telling the animal. That is, whereas a typical retinal ganglion cell, the center tends to win out over the surround, and you often do get some response. In a geniculate cell, you get far less of a response. So the tendency of these cells not to respond to diffuse light is something that is decidedly increases at the level of the geniculate. That's one thing it does. The lateral geniculate body also gets input from the brainstem, and the cells work better. They transfer the information up to the cortex in a much more effective way if the animal is awake than if it's asleep. And so it acts sort of like a valve that turns off when an animal is asleep. This is known to be so. Both these things that the geniculate does is known to be so in a cat. It hasn't. Neither one is known for sure in the monkey, probably simply largely because people haven't looked. Now, so much then for the way lateral geniculate body works, and maybe if we could have the first slide, which is a picture of the lateral geniculate. If I press this button, is there a chance that we'll get, if I get out of the way? Yeah.
I'll close the shades. And then the last button at the back there next to your left hand, Bill.
Yeah.
See if you do it manually, it should then hopefully get started. Did it turn itself off?
Oh, good. Yeah. So
Is that dark enough?
here, here's a picture of the lateral geniculate body.
Is there a mic, David?
Pardon?
He has one on.
Because we can't hear over the slide projector back here.
Oh,
Is there a
what would you like me to do? Shout or?
You might have to speak up a little bit.
All right. Can you hear me at the back now?
Well, I can now.
Oh dear. Yeah. You can turn off the. Anyhow, this is a lateral geniculate. This is the right lateral geniculate of a monkey. You'd have to be an expert to know that this was not a human lateral geniculate body. It is about the size of a peanut. So from here to here is a few millimeters. This is a cross section, a coronal section, which means that it's you've sliced the brain parallel to the face of the animal. So this is what a lateral geniculate body looks like. And the main thing to point out here is that it's a layered structure, 6 layers. And each layer gets its input from one eye and one eye only. So if you go back to the original picture that I drew on the blackboard, if this is the right lateral geniculate, if this is the left lateral geniculate, it's getting its input from the left half of the 2 retinas. So it is giving you information from the right side of the visual field. And it turns out that it, as a whole, it gets input from both eyes, but each layer gets its input from one eye and one eye only. So if I start at the top, this is the left geniculate. This layer is getting its input from the right eye, left eye, right eye, left eye, left eye, right eye. We don't know why the order reverses between these 2 layers. In fact, we don't know why there's an order at all, why the geniculate is arranged in this strange way. But in fact, no one really has the faintest idea why it should be built this way. But all of the cells in this layer get their input from the right eye, and it's topographically organized so that a certain region of the visual field will project, let's say, to this part of the geniculate. If you move over in the visual field, you move to a new region in the geniculate. And the topography is faithfully preserved all the way down. So if a certain part of the visual field activates these cells, the same part of the visual field via the other eye will activate these cells. And so that part of the visual field activates the cells in a column arranged in this way. So if you move your light in the visual field along, you're going to move the responsive cells over the layer like this in all of the 6 layers. So it's a topographic map. Each layer is a topographic map of the visual field, and the 6 maps are in register, in precise registers. So you stay in the same part of the visual field as you go from one layer to the next. But the take-home message here is that none of these cells gets input from both eyes. All these cells get their input from the right eye only, left, right, left, and so on. So the 2 eyes then are kept strictly separate at the level of the lateral geniculate body. So armed with that information, we can go on up to the cortex and ask what things are like there. And the lateral geniculate body in a monkey projects strictly to visual area 1 of the cortex. So I think the next slide. Can you hear me still at the back? Here is a piece of cortex. It doesn't matter the plane that this is sectioned in, but this is the surface of the cortex. It curls around like this, but you don't need to worry about that. The incoming fibers from the lateral geniculate, the axons, come in, streak up, and they land up in this layer here, this set of layers, which are collectively called layer 4. And these cells have their axons going up and down, so the information spreads to all the layers, but it starts out coming into these layers here. And so it turns out that if you record from these cells, the first thing you want to know is how do the cells respond to stimulating in the visual field. Typically, you have the animal anesthetized looking at a screen, and you shine patterns of light on the screen, and you ask, what can you do to influence the cells that you're recording from? So you do this in the geniculate, and you get the center surround receptive fields that I've talked about. You do the same experiment in the cortex, and for these cells in these layers, what you get is roughly the same as what you found in the geniculate. The cells have receptive fields that are center surround. And as you move along in the visual field, you move along one of these layers. By the way, the size of this is from here to here is about 2 millimeters. So if we take the entire length, this part of the cortex is about the size of a credit card, and its thickness is 2 millimeters. So if we were looking at the entire primary visual cortex, it would go over to the edge of the building probably. So just to give you an idea what the dimensions are, this is only a small part of the visual cortex you're looking at. And all the cells in this region are going to tend to respond to a certain part of the visual field. So the whole visual world is mapped onto the primary visual cortex. If you record from typical cells in the upper or lower layers beyond layer 4, where the fields are centered surround, you find that these cells are much more fussy about what they respond to. And a typical cell will respond almost always best to a line stimulus. That is a dark line on a light background, or a light line on a dark background, or a border between light and dark. Whatever kind of line you use, they respond when that line is in a particular orientation, not in a different orientation. That's the first thing that is almost universally true of the cells in the primary visual cortex, with the exception of the cells in the layer 4, where the fields are center surround. When I say fields, I mean receptive field, which, as you must know by now, if Bevel has done his work properly, receptive field is a region in the visual field, which when stimulated make the cell fire. If let's go to the next slide. Here is our responses from a typical cell in the primary visual cortex. This is the baseline of an oscilloscope tracing, and from here to here would be about 5 seconds. And what we have the animal looking at a screen, and onto the screen we have a line, a light line on a dark background that we're moving up and down, perpendicular to its direction. And you can see from this that when the slit is in this orientation, we get a response every time we move the slit from left to right, and no response from right to left. So this diagram shows you that the cell is selective as to the orientation that it responds to, and in addition, it happens to be selective to direction of movement, responding only to this movement, but not at all to this movement. Different cells differ in the best orientation, and they differ in whether they're selective to direction of movement. Some cells don't care whether a thing is moving to or fro. Other cells do care. And you see plenty of both types. So that's the first thing you can say then about cortical cells. They're orientation selective and often directionally selective. The other thing that you can say is that their responses, if you stimulate the left eye and then stimulate the right eye, trying one after the other, if you record from cells in layer 4, that is the input layer of the cortex, you find that almost without exception, the cells will respond to one eye but not the other. If you go to the subsequent cells above and below layer 4, you find that most cells, but not all, respond to both eyes. So if we can back up, what you find then, if you go push an electrode parallel to the surface, transversely is the term we use, in layer 4, you go from cell after cell, finding that they all respond, let's say, to the left eye. Then at some point, there's a sudden switch, and from there on, the cells all respond to the right eye. And you keep on going, and then you have a switch back to the left eye. And typically, you go about a half a millimeter along layer 4, parallel to it, and you switch eyes from one eye to the other. So that tells you then that this is a sort of a patchy thing. The cells, the eyes do not mix, as it were, in this layer. The connections from this layer above and below, the axons tend to bifurcate, and they go often straight up or often obliquely, so that when you come to cells in the lower layers or the lower layers, you find that typically cells respond to both eyes. And when a cell responds to both eyes, what you find is that the orientation selectivity is identical for the two eyes. Whichever eye you use, if one eye tells the cell to fire when a line is tilted this way, then the other eye will favor the same direction of tilt. So all that you find out when you stimulate one eye is identical to what you find out when you stimulate the other eye. What differs between the two eyes is the power of the eye to make the cell fire. A typical cell then will fire better, consistently better to one eye than to the other. And it depends on the cell. Some cells favor the left eye, others favor the right eye. Some cells, it's a draw, both eyes, the two eyes are equally effective in activating the cell. Some cells only respond to one eye, they're monopolized by that eye. And so you can imagine then, if you have fibers from this layer going every which way, a cell here will tend to favor the left eye if it's directly over a left eye patch. Another cell, if it straddles a left eye and a right eye patch, will tend to fire to both eyes. It just depends on where the cell is situated. Now this we learned in a few months recording from monkeys, and it suggested to us that these patches down here of left eye, right eye are about a half millimeter in length, but we didn't know whether the half millimeter was, if you were to stand up here and look down on the cortex, we had no way of knowing whether this would be like a checkerboard or whether it would be a lot of parallel stripes or a mixture of the two. And so we had to use anatomy to figure out what the answer was, how these things are arranged. By the way, a region where the cells respond only to the left eye here, and above and below, directly above and below, the cells respond to both eyes but favor the left eye, that we use the term ocular dominance column. to designate such a region. And the word column comes from the fact that the region favoring that eye will be sort of pillar-shaped. And you go 0.5 millimeter over and you come into a neighboring pillar where the cells all favor the other eye. And it's a monopoly in this layer, and the favoritism is it's a matter of where the cell sits relative to the patch that it's over. So if you're straddling a left eye, right eye patch in layer 4, if you're sitting up here, you'll tend to, the cell will tend to about equally to both eyes. Now, we had long wanted to know what these patches would look like if you were to stand up here and look down at them. We hit upon several anatomical methods of finding that out. But probably the most effective one so far involves injecting the eye right into the vitreous of the eye, injecting a radioactive amino acid, which is taken up by the retinal ganglion cells, made into a protein, and as is typical for nerves, the protein is shipped along the axon till it gets up to the lateral geniculate. And then if you take the lateral geniculate and section it and put the sections onto a silver emulsion like a camera film, the radioactivity turns the region of the film that it's close to, it turns it dark. So by developing the film, you can get a picture of the lateral geniculate. And just to show you what it looks like, if you inject the eye, here is the left lateral geniculate, this is the right eye. We've injected the left eye with a radioactive tracer, and these are the left eye layers. The radioactivity has come up to these layers, and the film turns dark. So this is called autoradiography. You may have heard the term. And of course, these layers and the other geniculate are the complement of the two. So this layer is the same as this layer. So this just shows you that you inject the left eye, and these are the 3 layers that are dark, whereas on the other side, these are the other three layers that are dark. So this is what I just told you about how the geniculate, how the architecture of the geniculate is arranged. This is sort of a proof of that, a demonstration of that using autoradiography. What we didn't know but hoped would happen was that the radioactivity would be eaten up by the cells in the region where it came and shipped down along the axon up to the cortex. And we had no way of knowing whether we'd get transneuronal transport, but we put in a whale of a lot of radioactivity into the eye that we injected and hoped for the best. And when we looked at the cortex, it turned out that we were sort of richly rewarded. And here is a picture of cortex. This is the top of the cortex. This is visual area visual 1, and the layer goes along like this and along like that, and this is white matter. So this is layer 4 along here, and that is where the geniculate fibers project. And you can see here are the patches having injected the right eye. These are right eye patches, and it goes right, left, right, left, right, left. So here was an anatomical proof of what the physiology had suggested. So these are the parts of the ocular dominance columns that involve layer 4. If you want to know what this looks like in 3 dimensions, these patches could represent a checkerboard, or could represent parallel stripes, or some mixture of the two. And to find that out, we take the other hemisphere and cut it parallel to the surface, do a tangential cut, and use the same technique and see what it looks like. Now, the cortex is slightly dome-shaped, so a given section might go through these layers and cut tangentially to layer 4 and pass out. Then a deeper cut would show this part and this part, still deeper, this part and this part. So you'd have to take serial sections and plaster one over the other and combine it to see what the whole of layer 4 looked like. But you'll see, that's probably not too clear, but here is a section that goes through layer 4, is deeper to layer 4 and superficial to it. And here are the regions that are stained. And so you can see that these are parallel stripes, and a cut in this direction would show a patch here, none here, a patch here, and none here, as in the slide I just showed. This is the other dimension, and this shows the cortex as though you were standing up above looking down at it. And by taking serial sections, some of it which will pass through this region, others will pass through successive rings further and further out, by mounting them in series, you can get a much wider idea of these parallel stripes. So if you injected the left eye, these stripes, this would be left eye, right eye, left eye, right eye. The radioactivity here shows up as light rather than dark because we're using dark field, which is a more sensitive way of using a microscope, and something instead of being dark looks light. But you don't need to worry about that at this point. This is the optic disc, and I leave it as an exercise for you people to figure out why the optic disc on one side in one hemisphere would look like this, and in the other hemisphere it would look pitch dark. So I won't plague you with explaining that. So now from what I've said, you know that if you sample a lot of cells in the cortex at random, you're going to find some that are equally influenced by the two eyes, some that favor the left eye, others favor the right eye, and the favoritism can be so complete that it amounts to a monopoly. So you may have cells that respond only to the left eye and only to the right. And so we decided to illustrate this by inventing a classification of cells. We divided our cells, every cell we saw, we put into one of 7 groups. And we said that a cell that is in group 4 responds only to one eye and not, it responds equally to the two eyes. Group 1 is a cell that responds only to the contralateral eye. So if we're in the left hemisphere, it's a cell that responds only to the right eye, not at all to the left eye. Group 7 would be a cell that responds only to the left eye and not at all to the right eye. And these intermediate groups are intermediate amounts of favoritism. So if you record from 500 cells at random and ask what group does each cell that you've recorded from favor, you can draw a histogram, and if this is the histogram that you find, which is typical for a cat, then this tells you at a glance that most of the cells respond equally well to the two eyes. A certain minority respond only to the left eye, others only to the right. Still others respond to the right, both eyes, but favor the right eye strongly or favor the right eye weakly. So you can, at a glance, you can sort of see what the balance is in the population of cells to the two eyes. So this histogram tells you right away, just at a glance, that there are a lot of binocular cells, that is, these cells in these intermediate groups. There are a fair number of cells that are strictly monocular, responding to one eye only and not the other eye. And there are a fair number of cells that respond about equally to the two eyes. So a glance is enough to tell you what the situation is in the balance of the two eyes. for a large population of cells. Of course, if you can find the cells that you recorded from to layer 4, you would find only cells in this group and in this group, because all the layer 4 cells are monocular, as I've said. The upper layers, though, this histogram represents mostly upper layer cells, and so this sort of tells you what the balance is. So you can see from what I've said right away that if you have brought up a kitten with one eye closed, then if after a few months with that eye closed you record from a whole bunch of cells and draw a histogram, you can tell right away what the preponderance of the two eyes is. And I hope that the next slide shows you. Here is a diagram. This is an animal where we closed the eye, one eye, for 2 weeks. I'm sorry, we closed one eye for 2 weeks. We closed one eye at 2 weeks of age. That for a cat is very young. The eyes don't normally open for the first 10 days. They're closed. So 2 weeks is very early in the cat's maturation. So here's a cat whose eyes have just opened, and we sew one closed and leave it closed for 18 months. And then we anesthetize the cat, open the eye that was sewn closed, and record from a bunch of cells in the cortex. And here we recorded from something like 100 cells and classified each one according to its ocular dominance. And you can tell right away that this is far from a normal histogram, far from looking like this. We find that most of the cells have come to be monopolized by the eye that was open all along. So to our great satisfaction in a way, here was we had no idea what the result of this experiment would be, and it turns out that it's a terrific result. And now, at this point, we can think of a lot of things to do to extend this, and all of these things kept us busy part-time for the next few years. Let's just go through some of the questions that you would like to ask about an animal like this. First of all, was it important that we sewed the eye closed at birth? Had we waited till until the kitten had become a cat at the age of 1, close the eye then for this period, and then open the eye and record, what would we find? And what we actually found was that it made no difference at all. That is, we hadn't produced an abnormal histogram. The histogram that you see with a late closure is a normal histogram. We hadn't influenced the balance of the two eyes. So then we could vary the timing. Some cats, we would only sew the eye closed at an age of a couple of months. Others wait until the cat was an adult and sew the eye closed and leave it closed for a year and ask, does it make any difference in these animals? And it turns out that the timing, you can sum it up by saying that you get these deprivation effects during a period that lasts 3 or 4 months in the life of a kitten. And if you don't do the closure until beyond that period, then you don't get these effects. So there is a period early in the life of the animal, which came to be called the critical period, during which you can get deprivation effects that are more or less marked. And you can vary all of these parameters, the length of time you leave the eye closed, when you close the eye. Having closed an eye, you can open it and leave the cat for a year and see whether there's any recovery. And to sum the recovery story up, it turns out that opening an eye will give you a recovery only if you open the eye during the critical period of the first 4 months. If you open the eye in an adult cat, you never get recovery. You never get back to a normal histogram. And beyond that, there's a curious story that you get much more recovery if after opening the eye that was closed for the first month or however long you closed it, you open that eye. If you close the other eye, that promotes recovery markedly. It all depends on how long the initial closure was. If you closed the left eye for 6 months, then whatever you do, you won't get much recovery. If you close the eye for the left eye for a few weeks, then in order to get a substantial recovery, you have to not only open that eye, but close the eye that was open. So that's called eye reversal, and eye reversal is an important thing to do if you want to get recovery. A lot of other questions came up. First of all, one of which is it light deprivation when you sew the eye closed, or is it form deprivation? You're obviously preventing any forms from getting onto the retina, and so cortical cells aren't going to respond because they only respond to contours. So that seemed to be an interesting thing to ask. And so in a few cats, instead of closing the eyelids, we covered the eye with a contact lens that had the consistency of a ping pong ball. So that then cut down the light to some extent, but it abolished all form. And the result of that was exactly the same as though we sewed the eye closed. So it's form deprivation rather than light deprivation that's producing this effect. Another thing we could do is look at the anatomy. We could in an animal deprived for 4 months, say, with a very abnormal histogram, we could look at the retina under a microscope, or the lateral geniculate, or the cortex, and see whether there were any changes. In the retina, we didn't find any changes, maybe very mild, subtle changes that you would have to use statistics to bring out. And if you have to use statistics, it means to us that you don't have a result. So in looking at the geniculate, it was enough to take the slide and hold it up to the light. You could see that you had made a marked change in the geniculate, whereas the retina showed nothing. Let's look at the results of the geniculate. And here, this is a rather pale stain. This is much better. And here you can see that the layers that got their input from the eye that had been closed, the cells are smaller than normal, these 3 layers, and they're paler. The normal eye, the cells are darker, and they're bigger. And you can measure the diameter of the cells, and we took months to do that and showed that there was a 20% difference in the diameter of the cells than the average. So in the geniculate, there are major changes. If you, this by the way is a Nissl stain. This shows only cell bodies. Every dot here is a cell body. The Nissl stain doesn't show axons or dendrites. So it's an easy way of telling that the cell body has become paler and has shrunk. Now, if you look at the cortex with a Nissl stain, you don't see any abnormality at all, which confused us at first. But it turned out that in the cortex, you have to use more sophisticated methods to see a change in the cortex. And we were pretty sure that there should be changes, because if you drive a microelectrode parallel to the surface through the cortex, and you're in layer 4, you find patches, left eye patches, right eye, left eye, right eye, and you can measure how far you have to push the electrode to get from one patch to the next. And it turned out that in a deprived cat, the size of the patches corresponding to the eye that had been closed was reduced. So we had the impression then that the ocular dominance columns had actually physically shrunk as a result of the eye closure. And I think that may be shown with any luck on the next slide. Yeah, what I want to, yeah, all right. Here is the tangential section through the cortex then, and these stripes are the eye in layer 4. This is layer 4, and these stripes are correspond to the eye that had been closed. So the open eye stripes are expanded, and the closed eye stripes are shrunk. So there's a clear change in the cortex. Even though a Nissl stain didn't show us anything, apparently the cells aren't abnormal, they've just become monopolized by a different eye. So there are major changes then in the cortex. The next slide shows, I think, this is a montage of many, and you can see again, these regions correspond to the eye that had been closed, these the open eye. So anatomically then there is a big change, and the ocular dominance columns corresponding to the eye that had been closed shrink. Now at this point you can ask somewhat more profound questions, and I don't have many minutes left. In fact, I have 1.5. But one thing you want to know is what is the nature of these abnormalities? You get a really abnormal cortex if you bring an animal up with the eye closed for the first month or so. What is this? What's going on? Is it disuse atrophy that causes the columns to shrink, for example? Is it analogous to putting your arm in a cast for a month, which causes the muscles to shrink in size just because they're not used? In order to get at that, we had originally done all these experiments sewing one eye closed, and we sewed one eye closed rather than sewing both eyes closed, just because we thought it was a rather mean thing to do to a poor cat, to blind it completely. But at this point, we figured that we had to get further, we had to look at a binocular closure, and we had no idea what we would find, if indeed we would find anything. So we brought up a cat for 3 months with both eyes closed, then anesthetized the cat, opened the eyes, and recorded from the cortex. And if you made a prediction based on what you found with monocular closures, what you would expect to find with a binocular closure is hardly any cell that would respond at all, because closing one eye gives you a cortex with hardly any cells responding to that eye. What we found, to our amazement, was that about 50% of the cells seemed to be responding more or less normally. And so we weren't at all finding the sum of the effects of closing each eye individually shut. So there we had a problem, and we had to conclude from that this is a matter of competition. With one eye closed, the other eye takes over, and you can see that the columns from the other eye expand, those from the closed eye shrink. So the open eye is taking over the territory of the closed eye, and if you close both eyes, there's no competition left, or much less competition, and you get a much more extreme result. So that was the first result. The second question we asked was, is this a question of depriving the animal of vision in the first 3 months? Is the result a matter of, in some sense, the failure of the animal to take advantage of those 3 months to learn to see out of the eye that had been closed? Or is it that the connections are all there at birth, and closing the eye is causing them to wilt, to become atrophic through disuse? The way of answering that was pretty clear, although it turned out that it wasn't very easy experimentally, because newborn animals are delicate. But by recording first from cats and then from monkeys the day after they were born, or soon after they were born, what we found was that in the case of the monkey, a macaque monkey, you would have to be an expert to know that you weren't recording from an adult monkey. We found everything that we had found in adult monkeys. We found the same thing qualitatively in newborns. That is orientation selectivity, directional selectivity, something called end-stopped, which I don't have time to go into, but a rather sophisticated result. So the conclusion is then that in monkeys, and probably in us, at birth, this part of the brain is wired up and all set to go. And in a way, I think that makes sense, because there'd be no reason why, if nature can wire a thing up so that it's ready to go at birth, it surely will do so and not make it necessary to acquire the connections by something process that's analogous to learning. In a newborn monkey is much more mature visually than a newborn human. And a human doesn't use his or her eyes for the first weeks. If you've seen, about the first month, if you've been around babies, you know that they're not using their vision a whole lot. A newborn macaque monkey is looking around and taking a keen interest in what's going on. It obviously is very much more mature. And I think that this is a matter of chance of how old the animal is when it's born. And you don't want a human to grow too big before it's born, because to get through the birth canal is a bit of a problem. With a monkey, apparently that doesn't exist so much, that problem. And certainly, for whatever reason, the monkey is a lot more mature visually. And so it may not be a surprise. The connections are all there, or substantially there. You can argue about whether there are minor differences between a newborn monkey and a monkey a month old, but there's certainly not major differences. If one could do the same thing in a human, I think there would be big differences between the animal at birth and between the human at birth and at 6 weeks, for example. But I think that's just a question of the maturing of connections that are going to develop anyhow. But to complicate the whole thing, it seems pretty clear that if a connection is there at birth and it isn't used, the results that I've been talking about all along here tell you that if you don't use the connections, they're going to wilt, in some sense of the word wilt. They're going to go to pot, and they have to be used in order to work. So this, finally, do I have another 5 minutes before you all have to rush off to dinner? I said to the managers here to Bevel that I'd try to get you to the dining room for supper. But I just want to talk about one more thing, because this bears strongly on what Sue Berry will talk about. This is the question of strabismus. And it's a complicated question, and she will tell you a lot more than I can, but in the time allotted. But what we thought an interesting experiment would be to cut an eye muscle to make an animal cross-eyed or wall-eyed, a newborn animal, first cats and then monkeys. So you have a monkey that at a couple of weeks you've made so that the two eyes look outwards, the two eyes can no longer converge. And you ask, what, if any, effect is this going to have on the wiring of the brain? And one reason we did these is that it's known that a cross-eyed human, if allowed to remain cross-eyed, may lose vision in one eye. This is called amblyopia, and it's a loss of vision that must involve the central nervous system, because nothing that we know of goes wrong in the eye histologically. The trouble is in the brain. But they develop, a situation develops where the vision deteriorates in one eye, and it may result in blindness. This is the commonest cause of blindness, in fact, is being cross-eyed or wall-eyed from an early age. So we made a bunch of kittens wall-eyed by cutting an eye muscle, and then asked, would we find an amblyopia? So after a few months, we took one of the kittens and tested its vision, first in left eye and right eye. You do this by putting an opaque contact lens over one eye and ask if the animal can get along with both eyes. What we found in this animal is that both eyes seemed to have perfectly normal vision, as far as we could tell. So this was a disappointment. We hadn't produced an amblyopia. And I remember we had 6 kittens, all wall-eyed, walking around the room. And we wondered, was this going to be a complete bust? We hadn't produced an amblyopia. What is the point in even recording from the cat? What could we possibly expect to find? But we flipped a coin and decided to go ahead. And we recorded from this wall-eyed kitten, and what we found was an absolutely amazing result, at least to us. And I don't know whether this, there is the histogram. So you can see that the binocular cells have practically disappeared, and we have two sets of monocular cells. These cells in group 1 responding only to the contralateral eye, and group 7 cells responding only to the ipsilateral eye, and very few monocular cells. A very abnormal histogram, which tells us that actually growing up cross-eyed had caused a major disruption of connections in the cortex. So this got us going on this, and it told us that being cross-eyed from an earlier age in a monkey, and almost certainly in a human, would cause this disappearance of binocular cells. Now, we know that binocular cells, among other things, give us depth perception. And to get depth perception from a binocular cell, if you think it through, I don't have time to spell it out right now, but if you think about it, a binocular cell may have the receptive fields in corresponding regions of the two eyes, in identical parts of the two retinas, but but that may not be the case. A binocular cell may be driven through one eye from a certain region, and from the other eye from a region that's either closer to the fovea or further away than is normal. In cats, most binocular cells have the receptive fields in identical parts of the two retinas, and exactly corresponding parts. But in monkeys, it's pretty clear that some cells have the receptive fields displaced inwards or outwards in normal monkeys. And if the displacement is inwards, then that cell will be influenced best by a stimulus that's further away than where the animal is looking. If the displacement is outwards, then it'll be influenced best by something closer than where the animal is looking. So you have to imagine, but not only do you have whole populations of cells responding to different orientations, but you have, at the same time, whole populations of cells that favor different distances from the animal. These are the cells then that are responsible for stereoscopic depth perception. And what this tells us, without knowing anything else, it tells us that you must have a huge number of cells in the cortex to look after all of these variables. For any given region of the visual field, you have to have all orientations, all degrees of ocular dominance, and all degrees of disparity, which determine whether the cell's interested in things at where you're looking or closer or further away. And of course, that's nice, because it's exactly what you have in your cortex is a huge number of cells. And until this sort of work was done, it was baffling to wonder why you should have 100,000 cells per square millimeter of cortex. What are they all for? And you can see that there are so many variables. Any given cell will have the best orientation, the best distance from the animal, a variation in ocular dominance, whether it responds to movement in one direction or the other. And you can go on and think of all the other variables that you might want to understand, such as, for example, color. So it's no wonder that we have to have a lot of cells to do the job. So I think that's probably it in the interests of their dinner. But maybe...
Maybe you take a few questions?
Sure.
Why don't we, if anybody needs to leave,
Yeah.
have David take a few questions. Martha Patton's work on the three-eyed frogs?
Oh, yeah, yeah. Yeah, 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 yeah.
How many of you know about Martha Patton's
very
three-eyed
interesting.
frogs?
I think that the three-eyed frogs incorporate the three eyes to generate disparities between the three eyes. I knew you didn't ask it.
I don't know how frogs work with three eyes, but at least you get three sets of columns, as I remember. Is that right? It's a long time since I've looked at Martha Patton's work,
On
but
the side where the extra eye was added. So normally there's two eyes and they have separate visual fields, so you don't have ocular dominance. But if you plant an extra eye on one side so that they share visual field, on that side you get ocular dominance columns. Bill was asking if on that side, in that visual field, that frog has stereo vision.
yeah. Yeah.
I think they did find stereo cells, yeah.
It reminds one of Margunka Sur's work where you rewire optic nerve fibers so that they go to the auditory cortex rather than the visual. And the auditory cortex develop orientation selectivity, if I remember right. So here's a cortex that has no reason to expect visual input at all. And the same thing happens as in the normal visual cortex. So I don't know. Do you know of that work? It's pardon?
Question right here.
I'll leave it to Bevel. Yeah.
We're talking about ocular dominance columns and how they shrank and expanded. Is there a difference in the actual number of cells that are affected by that? So if, say, a column expanded, is it incorporating other cells from the other columns into its dominance, or is it or are the cells just growing or multiplying? Or
Yes, there's, as far as we know, the question, did you hear the question? As far as we know, when an ocular dominance column expands, it just means that the cells which normally would favor one of the eyes has been taken over by the other eye. And there's no reason to think, and I think good reason to think, that it isn't so, that there are more cells adopting that eye. So the total number of cells probably stays the same. It's hard to be absolutely sure of that, but it is clear that it seems quite clear that, and I forget the reason why it's all that clear, but that the cells are being taken over by the other eye. And yeah.
Christina?
Considering the differences in the length of the critical period in different species, is that critical period proportional to the lifespan of the species?
Sorry, say.
Is the critical period proportional to the lifespan of the species, or is it
Oh.
related to their use of vision?
I think it's pretty clear that the critical period, it lasts longer in humans, and humans have a longer lifespan. Whether those things are intimately related, I just don't know. But you're quite right in the length of the critical period and when it occurs is different in different species. It's certainly different in the cat, where it would be the first month or so. In monkey, where it's 6 months, something like that. In humans, from clinical evidence, you'd guess that it's a year or so. But I think the actual length of time in the human is less clear. But so those things go parallel with the lifespan, but whether that's necessarily important, I don't know.
We were discussing in class difference if there's like one critical period for the magnocellular pathway and another critical period that slightly overlaps with the parvocellular.
I think they are slightly different, and I forget in what way, but I never thought the evidence for that was all that. It may, there probably is a difference, but it's you have to look closely, I think, to see it.
Maybe something like a cheetah might have a longer
Yeah.
magnocellular pathway to focus on.
I didn't say anything about the magnocellular, are the layers, are the two bottom layers after the break in the sequence. And the way the cells behave, especially with respect to color, is quite different in the two sets of layers. And this is in macaque monkeys and almost certainly in us. But exactly what the magnocellular cells do as opposed to the parvocellular, there are certainly differences. They act differently. But how to interpret this isn't all that clear. There are people who think that binocular vision, stereoscopic depth, for instance, is a matter of parvocellular, of magnocellular cells. I've never fully believed that, but it's something one can discuss and wonder about. I don't know how Hubel thinks about it. And, yes?
If you were to speculate wildly about Sue Barry, who gained stereovision late in life, about what would have happened if we had, prior to her gaining of stereopsis, put some electrodes in her brain, and then after she gained stereopsis, put some electrodes in her brain, what would you speculate you may have found?
I think it would be pure speculation, but I would think that in monkeys the result is so striking that the cortex consists of monocular cells. You saw the histogram. If you were to do a similar cell study in a cross-eyed human, I would think you'd find a similar thing. And after the recovery, after the training, if you regain stereopsis, that would be the most interesting question you could ask. Does that change? And I would think it probably does. So there must be some plasticity. Obviously, there has to be plasticity somewhere, otherwise she wouldn't have regained her stereopsis to the degree that she did. But whether that would be paralleled by the striking anatomical and physiological change that we see would be the first question you'd want to ask, and no one would be able to say for sure. I think you must end up in the brain with cells that are binocular and are responsible for stereoscopic depth. The fact that she can see it, I think it would be very surprising. Whether in the process of learning, what she has done is to develop new kinds of cells, or to get the cells that had wilted, get them back active, as though you were to take the cast off the arm, which gave atrophic muscles. One doesn't know, and one would certainly love to know the answer to that. My guess, I think, would be that the cells that have wilted, where the connections have become less effective, recover in some way. And the reason for the recovery is that she has learned to fixate. The reason why most cross-eyed people, even if you repair the cross-eyedness and do as good a job as you can, the best you can do is within a degree or so. And if having done that much surgery to get it within a degree, if then you do as the optometrist did with Sue Barry, train her and practice her on fixating so that the two visual fields are in register, then there's a chance apparently for recovery of stereopsis. And I think no one that, to my knowledge, had ever succeeded in that, and it was necessary to get accurate fixation before that could be done. This is just my guess, but I think she probably would agree with this. Yeah. Yeah.
It just would seem to me, though, that Sue Barry's situation is not directly analogous to the experiment that's being conducted in monkeys, because she does report an early experience of having had stereopsis.
Yes.
Which would presumably, if she's capable of having that recollection, have been post her critical period for wiring up stereo 2 neurons. So the analogous experiment would be to take a juvenile monkey and then make them strabismic, and then at some later stage realign their eyes and test to see if they have binocular cells. And my bet is that that's
You may
entirely
be right,
consistent
yeah.
with the data that you guys
I think
generated, you and Torsten generated.
here, what is not clear so far is whether, just as you're born, as a monkey is born with the cells wired up for orientation and directional selectivity, is the monkey born with the necessary connections for stereopsis, or is experience necessary to give that development? And I think the only experimental evidence is an experiment done by Ramachandran and Whitridge in sheep, who they recorded from newborn sheep and found that there were cells that had binocular disparity. And this suggests that the binocular cells are there at birth too, but they may have to be used in order to get them going. And there's always this ambiguity between training to learn something early on or whether the effects of training are to keep cells going that were hooked up appropriately at birth. And that isn't fully ironed out. But as far as I'm concerned, the evidence that we have all suggests that certainly the basic things, orientation selectivity especially, is there at birth. And if it's not used, it deteriorates. Whether connections for stereopsis are there at birth, we don't know for sure, except for the sheep experiment. I don't see the point in requiring experience for them to get wired up if they can be wired up at birth as the others are. But and it's a big, the nature versus nurture is a profound question, and there are people that take very strong sides, as we know. But if you read Pinker's books, for example, he's very much on the side of things being wired up, and Chomsky with language and stuff. But nobody thinks that we're born knowing the alphabet. And so whatever connections are necessary for us to read, we have to learn. There may be some underlying thing that's necessary at birth for us to be able to do that. But it's pretty clear that we found that our deprivation work almost in the 60s, where there were all sorts of political disruptions anyhow, tended to get us into trouble because every psychologist especially had looked on the newborn brain as a tabula rasa on which experience writes its message. And to find that anything is wired up at birth, as we did, was met with skepticism, and a lot of people wrote one paper after the other trying to show that we were wrong. And we did as we usually do. We just ignored it and went on our own sweet way and figured if we're wrong, that's too bad, we'll be sad. But let somebody else establish that we're right, and we can meanwhile do new experiments on something more interesting. I think that's the way to behave in cases of controversy, but I don't. It's just as well that we ignored the whole thing, because it evaporated for the most part, and people had to agree that a newborn monkey has orientation selectivity and so on. But it's
One more question, and then we should really stop for everybody's. Go ahead, Joanna.
I don't know if you've heard about the case in India that
I'm sorry. Yeah.
I don't know if you've heard about the case in India. This man who's blind and only sensed light and movement. Later, at 30 years of age, he was given glasses and was able to detect or see actual objects in color. And it was a pretty, and he improved. It was pretty miraculous. So I was just wondering what you thought might be happening there.
He was able to see color.
Color and see objects.
Yeah.
And distinguish objects.
I don't know. I didn't mention that one reason why we got started at all the deprivation experiments was what many psychologists, or a few psychologists, had brought up animals, including chimpanzees, in the dark. And when they brought them after 6 months or whatever the time was into the light, found that they were blind. And that suggested that we weren't surprised when our cats with closed eyes turned out to be blind. And what we wanted to know, what is the physiology and anatomy behind this? But it had already been shown that you can produce blindness by raising an animal in the dark. And the recovery from the blindness was slow and grudging and incomplete. But whether or not the best things were done to train the blind person in an attempt to regain vision, you don't know. And the thing that was good for Sue Berry was that she went to an optometrist who took an interest in this and taught her to fuse by doing appropriate exercises, and apparently it all worked. So you don't know a person who is dark-reared and turns out to be blind. It may be possible to get the thing working, but you have to do the right things, and one doesn't know. We know that bringing an animal up with one eye closed, you get better recovery, but still incomplete if you open that eye and close the other eye. And if you don't close the other eye, you don't get recovery at all. So that's one example, I guess, of having to do the right things. And a clinician to restore a defective sensory system may have to use a lot of imagination to hit upon the right things to do. Yeah.
I guess the bright side of all that is that there appears to be more plasticity than we would have expected a while back, at least in the human.
Yeah. The
training
problem
made a
was that
difference.
we never made any effort to correct a strabismus, to correct the cross-eyedness or wall-eyedness in our animals, because it seemed clear to us that we, with the best surgery in the world, you could never get the eyes placed properly so that within a half a degree or something like that, the two visual scenes would be exactly superimposed in the two eyes. So we didn't even try to look for recovery in our strabismic animals. What we needed probably was an optometrist specializing in monkeys who could train the animal to do that, but that we didn't have.
Thank you very much.
Yeah.
Rita.
Yeah.